So good morning to all. So we welcome you on the lecture number 20 of our Gyan course. So today lecture will be delivered by Professor Kavita ma'am, and the topic is about precision therapy exploiting synthetic lethality and case study. So thank you.
Thank you, Leknath. So we already discussed synthetic lethality before, right? So today I'm going to show you 4 examples of synthetic lethality. Number 1 is the famous BRCA1, BRCA2, and then we'll take 3 more cases, okay? So just to remind you, synthetic lethality means that, okay, it is our fault we started early, so I'll give you 1 minute to get settled. No, it's not your fault. We started early. We should not have started early. You guys are on time. It's 9:29, so just take your time, settle down. फिर से शुरू कर सकते हैं आप उसको ना रिवाइंड करके फिर से शुरू करते हैं. Lekna, you don't have to introduce me again, right? That can be edited later on. Okay, ठीक है. आप लोग सेटल हो जाए तब शुरू करते हैं. Settled? Okay, good. Okay. So I told you about synthetic lethality before. So today I'm going to give you 4 cases of synthetic lethality. Okay, and you guys know that very well. So for example, if there are 2 pathways for the same outcome, you delete one, the cells will use the other pathway and they will remain viable. Either A or B, you can delete either of them and the cells remain viable, but if you delete both of them, the cells, they die. So what happens in cancer cells, often one pathway is already defective, so they entirely rely on the other pathway, but normal cells have both A and B pathway. So if you inhibit the other pathway, normal cells will survive, cancer cells will die. Okay, so that is that. This is the same thing shown here. So these are called cancer vulnerability. We are targeting cancer's vulnerability because it is addicted to one pathway, while normal cells have two ways to achieve the same outcome. Okay, so this is exactly here shown here. Non-malignant cells, they have 2 ways to attain cellular viability. So either you delete this or gene A or gene B, it remains viable. But in tumor cells, one is already defective. So if you inhibit the other one, they die. Okay, and normal cells live. So you guys know about BRCA1, BRCA2 in PARP, but I'll briefly tell you once again, and then we'll discuss 4 other cases. So what are the functions of BRCA1 and BRCA2? They are tumor suppressor proteins, and they are involved in DNA repair pathway. Okay, so DNAs are broken in 2 ways: single-stranded break and double-stranded break. Double-stranded breaks are obviously much more complex and much more dangerous. So these guys are involved in repairing the double-stranded breaks, okay, by a process called homologous recombination, or HR. That is the function of BRCA1 and BRCA2, and they ensure DNA replication happens with minimal errors. DNA is our hereditary material, so cells make all efforts to make sure there are no mutations introduced. Okay. So now part 1 is also a DNA repair enzyme that identifies and repairs DNA damage, primarily single-stranded. Okay, so if you inhibit PARP, then BRCA1 and BRCA2 takes over and they will repair it. Okay, so PARP under normal circumstances will repair single-stranded DNA. BRCA1, BRCA2 will repair double-stranded DNA. But if you inhibit PARP, BRCA1, BRCA2 will take care of both single-stranded and double-stranded. If you inhibit BRCA1, BRCA2, PARP will take care of both repairs. So they compensate for each other. But in many cancers, BRCA1, BRCA2 are deleted because they are tumor suppressors. So cancer cells have a habit of getting rid of tumor suppressors. Okay, so they get rid of BRCA1, BRCA2, then the cells are entirely dependent on PARP to repair DNA damage. Okay, and in that case, if you inhibit PARP, normal cells don't care because they have BRCA1, BRCA2, but tumor cells will die. That is synthetic lethality. Okay, so this is shown here. Single-stranded breaks are fixed by PARP, and if you add PARP inhibitors, then this single-stranded thing will not be fixed. Eventually, this will lead to double-stranded break. So normal cells will fix it because they have BRCA1, BRCA2, but cancer cells will die. Okay, so just to give you an idea, in breast cancer, 50 to 60% of breast tumors have BRCA1 deficiency, 40 to 55% have BRCA2 deficiency. Males have very little deficiency of BRCA1 in the breast cancer. Okay, pancreatic cancer, 1 to 3%, both male and female. Ovarian cancer, again, huge percentage. And prostate cancer, also some percentage. Okay, so at BRCA1 and BRCA2, they are on different chromosomes here. Okay, so BRCA1, BRCA genes, and so BRCA1, BRCA2 deletion is pretty common in cancer. Okay, and when people have BRCA, yeah, go ahead.
This PARP enzyme, so when the single strand breaks, is it the diester bond that breaks, or which one breaks, and then how is this located? Which one is broken?
So there are many ways for DNA damage to occur. So sometimes what happens that this is the whole strand of DNA, right? So phosphodiester bond remains intact. The purine ring will be hydrolyzed. So it's like a missing tooth. The gum is there, the tooth is missing, right? So purines are removed very easily, 5,000 times per day per cell in our body. Pyrimidines are 500 times per day per cell. So just removing the base, the glycosidic bond is hydrolyzed. That's very easy. But the phosphodiester bond is intact. Okay. So in that case, what they do base excision repair. So they come, they cut basically the gum from both sides just exactly. So this is missing just exactly next to it, fix it with a new base using the second one. In case of PARP, it is the phosphodiester linkage which is broken. So it could be one or two pieces, it could be a bigger piece. So in that case, they do nucleotide excision repair. So they cut out about 12 to 13 base completely along with the gum, right? Completely, then the other strand is there. So that acts as a template, and DNA polymerase 2 comes in and fills it, and then it is ligated. So that's how it works. Yeah. Prostate is unique to men, ovarian is unique to women, and in here, men also get breast cancer. So only 1 to 2% have deletion of BRCA1, up to 9% of BRCA2. But prostate is obviously only for men, ovarian. Just see the higher percentage. So women who have BRCA1, BRCA2 deficiency, they are prone to both breast cancer and ovarian cancer. That's how that actress Angelina Jolie, she had this ovarian fallopian tubes all removed. Okay, so this is a very easy test to get it done and be preemptive about it. Okay, so so this is really there environmental risk also with impaired BRCA1, BRCA2, because why environmental risk? Because environmental can break your DNA, right? If you're exposed to wrong things. chemicals or extremely bright sunlight for very long. You know, these all things lead to DNA damage. BRCA1, BRCA2 are absent. That is a very serious thing. Okay. And even long bouts of being sedentary correlates with lower BRCA1 gene expression. Okay. So when BRCA1, BRCA2 is lost, then homologous recombination repair fails. They cannot fix double-stranded breaks. Cancer cells become dependent on backup repair pathway, and that is where PARP-mediated things come. So that is why PARP, hey, no cell phone. Okay, no cell phone in my class. Okay, so PARP inhibitors, so if you use PARP inhibitors like olaparib, then DNA damage accumulates and the cells will die. And normal cells will live. Why? Because they have the backup pathway. Okay, so that is the whole idea of that. So these are FDA approved PARP inhibitors. Okay, and they are extremely useful. Okay. Now let's look at second case for synthetic lethality, KRAS and TBK1. So yesterday we spent a lot of time on KRAS. KRAS is one of the most mutated gene in humans, and it is responsible for vast majority of cancer, especially highly aggressive cancer. Now here is the thing, when these oncogenes promote cancer, they mess up the physiological signaling pathways to an extent that they become highly dependent on alternate pathways. Like somebody is doing the wrong thing, like really wrong thing, they always will have some helper who is a partner in crime, right? So that's what KRAS does. KRAS just stresses the cell so much because of uncontrolled proliferation and other things that it becomes highly dependent on a kinase called TBK1. So in case RAS dependent dependent cancer, it really depends on TBK1. So if you inhibit TBK1, it will die. It is that dependent. Okay. All right. So let's look at it. So we looked at this mutant last time, yesterday, G12C mutant. So this, remember, we discussed the source is activating KRAS. So KRAS, once it is activated, it turns on many signaling pathway. However, this is KRAS dependent cancers are highly dependent on TBK1 driven survival signaling. So if you inhibit TBK1, the cells will die. So this is just showing you RAS signaling pathway, which leads to proliferation, survival, migration, angiogenesis, DNA synthesis. And I showed you this yesterday. Look at the high percentage of KRAS mutants, right? These are, pancreatic cancer is extremely aggressive cancer, and 73% of pancreatic cancer have these mutations. So this TBK1 is a serine threonine kinase. Remember I told you there are three kinds of kinase, serine, threonine, tyrosine, and dual kinases, which can phosphorylate both tyrosine and serine threonine. So TBK1, for our thing, just remember, it is involved in cell survival signaling. And KRAS dependent kinases, cancers are highly dependent on TBK for survival signaling, compensatory pathway activation. So if you inhibit TBK1 in KRAS dependent cancer, these cancers will die. These tumors will die. Normal cells will not die because in normal cells TBK1 is not that important. But in KRAS, KRAS is the main guy and it depends on TBK1. So just inhibiting TBK1 will kill those cells. So this is a very good example. KRAS mutant cells require TBK1 for survival. So blocking TBK1 selectively kills KRAS mutant cells. Okay. So this is, you don't have to remember anything, but it's just showing you that it, just look at that, all the pathways. This is not important for you, but look at that. It provides inflammation. Cancer loves inflammation. Okay, it inhibits apoptosis and necroptosis. It promotes proliferation, survival, autophagy. Okay, so this is just summarizing the whole thing, that oncogenic KRAS usually have these mutations, and through RAL-B, it actually becomes highly dependent on TBK1. And TBK1 helps survive through anti-apoptotic pathway and FKB activation. So KRAS mutation depends It creates oncogenic stress, proteotoxic stress, metabolic stress, and for overcoming all these stress, it depends on TBK1. So if you inhibit TBK1, these cells die. So that is a very good example of synthetic lethality. Now let's look at MTAP deletion in PRMT5 MAT2A. So there are three players here. Okay, so there are three proteins that we have to consider: MTAP, PRMT5, and MAT2A. So I will tell you the functions of each of them, and then we'll see how they crosstalk with each other. So MTAP is methylthioadenosine phosphorylase. Okay, do you guys know what phosphorylase enzymes do? How do you define phosphorylase enzyme? Anyone? No, that is phosphatase. Yeah, adding phosphate group, right? So how do you so kinases also add phosphate groups, right? So how do kinase and phosphorylase enzymes differ? Anyone? Probably you guys don't know that, right? So kinase always use ATP to phosphorylate a protein. Phosphorylase use inorganic phosphate to add the phosphate group. That's the biggest difference. Write this down. This is important. Okay. Kinases use ATP to phosphorylate something. Phosphorylase adds inorganic phosphate to add something, to add the phosphate group. Okay. So what happens is that this enzyme MTAP, this is involved in methionine salvage pathways. That means it is involved in the synthesis of methionine. And this is MTA, which is 7-methylthioadenosine. It is converted to methionine in 6 steps. You don't have to remember the 6 steps. I have shown the 6 steps here. So MTAP is used in the first step, and then there are other enzymes. The point is that MTAP is converted to L-methionine, and it is, sorry, not MTAP, MTA is converted to methionine via NTAP. That is the first step of the reaction. Okay, and MTA is 5-methylthioadenosine. So that's one player you have to remember. This is the function, that this is involved in methionine formation. Then there is another enzyme which takes this methionine and it converts it to S-adenosyl methionine, or SAM. So this is the reaction: methionine plus ATP plus water. This enzyme converts to S-adenosyl methionine. Look at this structure here. This is S-adenosyl methionine. Nobody calls it S-adenosyl methionine. We all call it SAM. So this SAM, look at this methyl group, and this sulfur is positively charged. So this is a very good donor of methyl group. So whenever you have to do methylation in cells, this is the donor for methyl group. Okay, and so when it donates methyl group, it becomes S-adenosyl homocysteine, and then it is further converted. We will not go there. So once again, MTAP is involved in methionine formation. MAT2 is involved in SAM formation from methionine. Now PRMT5 is a methyl transferase who uses SAM to put the methyl group on different substrates. Make sense? Now this is PRMT5. So P stands for protein, R stands for which amino acid is denoted by R? Arginine. So this is arginine methyltransferase. Okay, so PRMT5 uses SAM because it is a methyl donor. And look at arginine side chain. It adds the first methyl group, the second methyl group here, right? It could be symmetrical or it could be asymmetrical addition. So now you see three players involved in the same pathway. One guy is involved in methionine formation, the other converts methionine to SAM, and the third uses SAM. Okay, so that's how the three proteins are linked together. So let's look at the synthetic lethality here. So what happens is that MTAP gene is deleted in 15% of all human cancers. Now here is the funny part. MTAP is not a tumor suppressor. So why would tumor cells delete MTAP, right? So actually they are trying to delete CDKN2A, which is a tumor suppressor. MTAP is just next to it. So when tumor cells delete CDKN2A, they accidentally delete MTAP also. So that is like just by being in the proximity, that also gets deleted. So what is the consequence? So this is called passenger deletion. Tumor cells don't want to delete it, they just accidentally delete it. Okay. So now what happens when MTAP is present, MTA is converted to methionine to residual PRMT5 activity. So maximum activity is inhibited by MTA. Whatever little is left, the cells are completely addicted to PRMT5 activity, and that's when you inhibit it and the cells will die. Okay. And the other option is that you inhibit MAT2A. So MAT2A is responsible for the synthesis of SAM. If there is no SAM, PMT5 cannot use it. Basically, it has the same impact. Are you guys following me? So what you do in MTAP deleted cells, PRMT MTA levels increases. PRMT5 is inhibited, almost inhibited. A little bit more push, they die. Just a little bit of more PRMT5 inhibition, they die. Or you inhibit this, so there is no SAM. So it's basically killing PRMT5, because without SAM, PRMT5 cannot function. So that is also synthetic lethal. Okay. And now we look at the, so this is exactly the same thing written there. Okay. So indirectly killing MTAP null cells by starving PRMT5 of SAM. Okay. So same thing written in details here. And these are just to tell you that keep in mind, MTAP deletion is accidental, right? They are trying to delete a tumor suppressor. Still, it is deleted in glioblastoma, pancreatic cancer, and these other cancers. Okay, and these inhibitors are there. Okay, so MTAP deletion alone, cells survive. If you inhibit MAT2A or PMT5, somehow they will tolerate it if MTAP is there. But if MTAP deletion is there, you inhibit this, it will lead to cell death. Okay, now the last one. My favorite kinase, aurora kinase. Okay, so aurora kinase is present in every cell of our body which is dividing, and it is an essential kinase for mitosis. And if you inhibit aurora kinase, the cells will die, normal cells will die. So inhibiting aurora is pretty toxic, but it is overexpressed in every known human cancer. So that is why it is also an extremely important cancer target, right? So this is just showing you it is involved in many different pathways here. Okay, and there are many substrates of aurora known. So there is a tumor suppressor called retinoblastoma. And if you have to guess, what do tumor cells do what do tumor cells do to tumor suppressors? They whenever there is a tumor suppressor, what would tumor cells do? They will get rid of it. Anything which stops tumors from going, they will get rid of it. That's how poor MTAP gets deleted. It's not even a tumor suppressor, it is just next to a tumor suppressor and gets deleted. So anytime there is a tumor suppressor hanging out, tumor cells will delete it, right? So they delete another tumor. Aurora is a kinase. Aurora is an oncogene. So that is a driver of cancer. So they will not delete that because that is driving cancer. Aurora is the master regulator of cancer. But cancer cells will delete tumor suppressor, and one such tumor suppressor is retinoblastoma protein. So when retinoblastoma protein is deleted, it leads to tumor initiation, proliferation, epithelial to mesenchymal transition. You guys know how bad it is. Genomic instability, it makes cancer stem cell. Remember I told you about cancer stem cell. You give very toxic therapy to a tumor, right? And 99.99999% cells die. Actually, one cell lives, just one cell. If that one cell lives, that is the cancer stem cell. And that one cell can form a whole tumor again, a very aggressive tumor, right? So cancer stem cells are very bad, okay? So if retinoblastoma protein is lost, it leads to cancer stem cell formation. So how do we combine Aurora A, which is an oncogene, to retinoblastoma protein? So retinoblastoma protein is obviously a tumor suppressor, and its role is in the cell cycle. and it really helps in the mitotic spindle formation. I'm telling you in a very generic way, very simple words. So it is really involved in mitotic spindle formation. Okay, and if retinoblastoma is lost, then the whole system is very fragile. Now, as long as aurora is there, it will take care of the mitotic spindle and the cells will survive. But in such cells where retinoblastoma is missing, if you inhibit aurora, they will die immediately. And that is what is synthetic lethality here. Make sense? So that's what it is saying. So primed spindle assembly checkpoint. Okay, so RB1 deficient cells have a hyperactivated or primed SAC. So now they are highly dependent on aurora A to fix the problem. And if aurora is even inhibited a little bit, the whole thing will collapse and the cells will die. Okay, and the cells will. So RB loss leads to replication state, chromosomal instability, dependence on aurora A for mitotic fidelity. If you inhibit aurora A, the cells will die. Okay, so this is just telling you different drugs of aurora A, and so these are pretty aggressive cancer, triple negative breast cancer, small cell lung cancer. Okay, almost 100% of these cases have RB1 mutation. So loss of tumor suppressor is a really is almost a hallmark of cancer. Okay, neuroendocrine prostate cancer, remember this is the latest man-made prostate cancer. They also have retinoblastoma, they also lose retinoblastoma protein. Okay, so this is just telling you, so one of the problem with aurora inhibitors are that they are pretty toxic. So recently this compound was made, LY compound, and this is 1,000 times more selective for aurora A than aurora B. Aurora and aurora B are like brothers, very similar to each other, not cousins, brothers. Okay, so very similar. So to make an inhibitor which hits only one and not the other is a big thing. Okay, so people think this may get approved by FDA. So this is just showing you, this is the SAC, remember? SAC is spindle assembly checkpoint. So if you have RB loss, this is defective, and now it really depends on aurora A. If you inhibit aurora A, completely the whole thing falls apart. So that is what is shown here, aurora and retinoblastoma synthetic lethality. Okay, everything I told you. All right, so the first lecture ends here. Second lecture and fourth lecture are very long. First and third lecture are short. So let's start the second lecture right away. Okay. Because now we will go to immunotherapy. How many of you have background in immunology, like pretty good background in immunology? 2, 3 people, right? 1, 2, 3 people. So most of you don't have it, right? So I decided to actually give you a crash course on immunology. What are immune cells? And because we have to know, we keep saying antigen presenting cells, but what are these cells, right? Exactly how they are presented, how do B cells form antibodies, which cells form antibodies. So you guys have to have some background of immunology before you understand immunotherapy. Right now all we are doing is PD-L1, PD-1, right? But we don't know what is what. Okay, so let's look at, so this word was coined by Bob Weinberg, crowd control. So what is happening? Cancer is crowd on top of crowd, right? So Bob Weinberg is the person, when I started this lecture, I showed you that thing, right? Hallmarks of cancer. That picture was made by Bob Weinberg. He's one of the leading scientists in cancer field, right? So he's the one who used this word, crowd control, tumor immunology, and tumor immunotherapy. So today I will tell you about these 3 phases. So the immune system is the one which saves us from cancer because it is always surveying it, and anytime something goes wrong, it eliminates the cells. So you guys may not know this thing. So what happens is that there are, I don't know how many cells in the body. So immune system, it is like you live in a society where the police comes and checks with you every time. You have to open the door and you have to show that you are all right and there is nothing there. And the police will check that nobody is hiding somewhere something. So how does immune system checks all the cells? So all cells which have nucleus, they have this ability that they will lyse their own proteins, not to amino acid, but to like 10 to 15 more amino acids, right? And they all will expose using a receptor called MSC1. They will expose it like this and show it to immune cells. These are our peptides. So the immune system will check the peptides. No foreign peptide, you are good to go. If it finds any foreign peptide, it will kill that cell. So every cell has to present themselves to the immune system. That kind of surveillance happens. So every cell of our body is constantly showing it to immune system, I'm okay, I'm okay. It's like opening the door and showing the inside of your home. Okay, here they are presenting it in that receptor. Please check it, we are okay. And if there's anything, it's gone. That's how we survive. So this receptor is MSC1 receptor. Makes sense to all of you? Okay, so that is what is shown here, that here the immune system is always surveying and eliminating that. When cancer cells happen, there is a constant fight, okay? So then there is some kind of equilibrium established. This is the best phase, elimination of all the foreign things. Here, basically the system is tricking immune system by PD-L1, PD-1 kind of thing. And then after a while, cancer cells are so smart, it is like you pay the police who comes to check your thing are becoming, if they are bad people living, so basically they bribe the police and police is helping the bad people. So the tumor cells basically trick the immune system, and immune system in fact helps grow the tumor then. So immune system is not only not working, it is also helping the tumors to grow. So it goes through these three phases, and all threes are E, triple E, okay? Elimination, equilibrium, escape. Tumor cells escaping and they become pro-tumorigenic. The immune system is helping, okay, to grow the tumor. So let's, yeah, go ahead.
Actually, told like the system works accordingly in which the antibodies keep inside it to check whether everything is alright inside the nucleus.
Not antibodies. Not antibodies. Antibodies are different things. So what happens is that, so suppose all nucleated cell, all the cells in the body which have nucleus, okay, so they have to digest part of their protein and keep presenting it. It's like a receptor, MSC1, not 2, 1. So they present these peptides like that, okay? And these T cells are the one, not B cells. They are the one which are checking that this is correct, this is all these are correct, and there is no, but suppose there's some viral infection and they show up, then those cells will kill that particular cell. So it has nothing to do with antibodies. Antibodies have not come into picture as yet. They will come later on.
Why not like RBC, because they don't have the nucleus and like
RBC is not involved in this function. So RBC is not, all nucleated cells, not RBC. Yeah. RBC, their function is pretty different from all other cells. They are oxygen carrier, they are involved in that, and then they will die after 120 days. So RBCs do not present MSC1. It's all nucleated cells. Make sense? Okay.
The people binds with these peptides to check whether it's okay.
Yeah, they are being surveyed. So anytime they see a foreign thing, that's when, so tumor cells are also nucleated cells, right? So they have to also show the MSC1, and they have these special antigens there. So they will be caught right away. So what do? They downregulate MSC1, so they don't even show that. So that's how they evade the surveillance from the immune system. Make sense? Go ahead.
so there's just a follow-up question, because since you said that RBCs are not involved in this whole machinery,
Yeah.
so how do we detect leukemia using the same machine? We cannot, right? Or can we?
So what happens is that when cells become cancerous, right? When cells become cancerous, they actually have their they are called neoantigens. They make their own antigens which are not present the in normal cells. And if those cells lice up, those antigens will be exposed. So then the tumor cells will become very active in cleaning up those kind of things. So there may be some additional ways to identify, because it is also possible that they will show the antigens on the top of the cell, right? So like HER2 is also an antigen, right? So tumor cells have two kinds of antigens. There are neoantigens and then there are HER2 kind of receptors which are overexpressed. So there are many ways for immune systems to detect the details of, like if RBCs are becoming really weird, then immune system cells will pick it up. Make sense? Yeah. Okay. So this is a very long lecture. Okay. So I will tell you about the immune cells in the immune system. So this picture shows you all different places where immune system, these are the places where immune system is active throughout the body. For example, skin is the one which protects us from outside things. If skin is completely intact, a lot of things will not get inside. Mucosa, like in our respiratory system, right? Lot of germs are trapped in the mucous membrane. So that is also considered part of the immune system. Tonsils have some kind of immune cells. Now, spleen is a very big place where WBCs are stored also. Okay, spleen. Lymph nodes are really important. I will talk to you about lymph nodes a lot. All the green things you are seeing here, it's not showing you the complete lymph nodes, but this whole lymphatic system from the head to the toe, it's completely spread out lymphatic system, and it's about every adult human being has about 500 to 700 lymph nodes. And lymph nodes are like, colander. They keep filtering the blood all the time and cleaning up if there is any infection, they will just clean up the whole system, okay? And they will let go of the nutrients, but they will catch any infection there. Okay, and that is why if cancer spreads to lymph nodes, it is really bad because lymphatic system is going throughout the body. So that's why it is considered very bad. Okay, and white blood cells include neutrophils, macrophages. We'll come to that in just a few minutes. So, so I was trying to put all the immune cells in one place. So it's very complicated because there are many classifications, but if we take all the immune cells, not the lymphatic cells, we are talking about the immune cells, right? So all the immune cells, they originate from hematopoietic stem cells. Tomorrow's lecture is all about stem cells, and you will come to know about hematopoietic and mesenchymal stem cells. So these are present in bone marrow, and there are two main lineages, lymphoid lineage and myeloid lineage. Okay, so to make it easy, I have made this green, I have made this red. Now, without going into really great details about everything, I want you guys to have these notes so that if you want details, you can always go back and refer to the notes here. But here I'll go quickly, okay? Because otherwise we'll get stuck here. So lymphoid progenitor cells make three kinds of cells: T cells, B cells, and NK cells. Okay, once again, lymphoid cells make, I'm going to skip here, they make lymphocytes. Lineage makes three kinds of cells: T cells, B cells, and NK cells. B cells are the ones which will make antibodies. Okay, there are many kinds of T cells, there are also many kinds of B cells, and then there is NK cell. So now I'm going to show you a little bit more details. So T cells are made in bone marrow, but they mature in thymus, and because they mature in thymus, that's why they're called T cells. T comes from thymus. And there are different kinds of T cells. So just to give you a quick idea, CD8 plus cytotoxic T cells, they will kill virus infected. So if you go and put your MSC1 out there and it detects virus, these T cells will just immediately kill it. Okay, So, and then CDK4 helper T cells, they help B cells, and if they help B cells, they make the antibodies. We'll come to that later on. These guys are TREGs. TREGs are actually immune suppressing cells. So these are immune promoting cells. TREGs are there. So normal body has TREGs to prevent autoimmunity, but cancer cells invite TREG for immunosuppression. Okay, so TREGs will play a very important role. And anyone can tell me what are memory T cells?
Cells which keeps the memory of previous infection.
Previous infection forever. So when we are infected with something, we never get infected with that because we keep memory of that all the time. Now, B cells are B lymphocytes. They originate from bone marrow as well as they mature from bone marrow. So look at the difference. T cells are also originating from bone marrow, but they are getting mature in thymus. But they are B cells, both origin and maturation is bone marrow. That's why B cells. And there are 4 kinds of B cells: naive B cells, memory B cells, plasma cells. So B cells mature to plasma cells, and these are the cells which make antibodies. Okay. And then Bregs are again similar to TREGs. We'll not discuss Bregs today. And then lymphoid cell is NK cells. These are natural killer cells. They see something, they eat it, they kill it. Okay, so these are the 3 kinds of lymphocytes. So remember I told you this is lymphocyte original. Now let's go to myeloid progenitor. So there are many of them. So without going into, so first you divide the myeloid into 3 categories here: granulocytes, mononuclear, and myeloid MDSCs. Granulocytes have granules in it, if you see. Okay, that is why these guys don't have it. But we will not go to granulocytes or mononuclear or myeloid. We'll just look at what are these cells. Okay, myeloid. So neutrophils, eosinophils, basophils, mast cells, monocytes, macrophages, dendritic cells. Okay, what is so special about dendritic cells? Can anyone tell me? Immunology students. Okay, you also add to that.
Dendritic cells are mostly in neurons, like they are profoundly, their function is to carry on the signal transduction.
That is, if I have to avoid any question, I would say they carry signal transduction because everything is signal transduction. It's like humans, like, a generic thing. You. Go ahead. You said MSC2, but you said it in the wrong way. They are the one who have MSC2. Okay, so you guys are hearing me say APC, 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 antigen presenting cells. These are dendritic cells. Dendritic cells are the gold standards APC. They present antigen. So what they do, if there's a viral infection, dendritic cells will take up the virus, chew it up, the peptides, make about 20 mer peptides, 13 to 20 mer peptide. And instead of MSC1, they have MSC2, which is slightly different from MSC1 in looks. And they present it. And then the T cells will recognize this is wrong. And that's when the whole immune system will start. So they are the famous APC cells we see, APC, APC. These are the guys. Okay, some other cells can also function as APC, but these are the main professional APCs, right? Okay. And then MDSC, I'm going to spend a lot of time on these guys. These guys are recruited by tumors and they suppress the tumor microenvironment. That's why tumor can grow. So they suppress the immune system. Okay, so this is just a quick summary of the whole thing. Lymphoid, T cells, B cell, NK cells, and these all come under myeloid lineage. Now, you guys have heard white blood cells. So what exactly are white blood cells? So this is yet another classification. So white blood cells include neutrophils, eosinophils, basophils. They also includes B cell, T cell, natural killer cells, monocytes. They all come under white blood cells, these things. So some of them are shown here. Okay, these are just white blood cells here. Okay, so they kill bacteria, kill parasites. So many times somebody is given cancer therapy, chemotherapy, especially chemotherapy, white blood cells also die. Okay, and then the person becomes very prone to infection. Why? Can you tell me why chemotherapy leads to less white blood cells? Anyone? So what is the principle of chemotherapy? Kill rapidly dividing cells. And where are these cells produced? Bone marrow is? That is the reason. You guys all got it, right? So these are suppressed. So I just told you, right? Lymphocytes give rise to T cells, B cells, and NK cells. And white blood cells are these cells here. Okay? So now let's look at B cells and T cells, the lymphocyte, because these are the guys which are involved in antibody formation. So there are mainly two kinds of T cells. We'll not talk about TREGs. TREGs are the tumor suppressive cells. We'll talk about those later on in the lecture. But right now we are talking about physiological stuff. So this is T cell. There are two main kinds, T helper cell and T killer cell. T killer cell will immediately, they spray material on the stuff, the cell. They spray perforin, granzyme, and they kill the cells. They shoot the liquid and the cells die. Perforin will form holes in the cell. Granzyme will go eat up the whole thing and that they will die. So that's the function of T killer cell, as the name suggests. T helper cell helps the B cells, and once they get the signal from T helper cells also, then they become a plasma cell and they secrete antibody. Okay, so we'll come to that in just a few minutes. So that's not the whole immune system either. Okay, so we'll come to antibodies which are produced by B cells. Then cells also secrete cytokines, and by secreting cytokines they can launch the immune attack. So almost all immune cells and many structure cells can produce cytokines. Okay, and these cytokines can immediately, depending on the conditions, when cytokines are released, they can regulate inflammation. Inflammation happens when immune cells are warding off invaders or healing damage to tissues. So if there is an injury, this immune cells will come, they will secrete cytokines. Why do they secrete cytokines? To recruit other cells to repair it, but that will also lead to inflammation. Complement system. What is a complement system? It's not a cellular system. It's a very important immune system also, which is composed of about 30 different proteins, not cells. These are just proteins, okay? And they are mainly made by the liver, surprisingly. So liver cells are the ones which make the complement proteins, and they are also they also constitute our immune system, okay? They help us. Now, lymph nodes is part of the lymphatic system. So look at this picture here. I think I have one more picture here. So do you see all these small bean-like structures? These are lymph nodes. Okay, so these lymph nodes are the ones. So lymph nodes are small bean-shaped organs like colanders to filter stuff. They filter waste products from the fluid that drains from your tissues and cells while keeping the good components like nutrients. We have about 600 on an average lymph nodes. Okay, they store and activate B cells and T cells. Okay, so this is a very good view. Look at the lymph nodes here. Okay, so that is why they are so there are 600 of them. So that's when cancer is there. If it goes to lymph nodes, then it can just spread very quickly. So usually when doctors are doing surgery, they will remove all the lymph nodes in the vicinity to prevent the cancer from accessing them. Okay, so it is just giving you some idea. In the neck they are 60 to 70, in armpit they are 20 to 40, and so on, it goes on. Okay, the exact number varies between individual, but about 600 in all of us. Okay, so this is, you can see, it is more detailed picture of lymph nodes. It's really heavy in this area here. So it maintains fluid balance, supports immune surveillance. Okay, So it is a one-way drainage system flowing from tissues, lymph nodes to the venous blood, and venous blood then is that purified. You guys know that, right? Now, in addition to that, there are other organs as well. So spleen is one of them. So spleen stores white blood cells that defends the body from invaders. It also filters the blood, recycling old and damaged cells to make new ones. Tonsils located in the throat, they can keep invaders as soon as they enter your body. Is tonsil removal common here in the absence of any disease?
Only if somebody has
Infection, right? In US, it was like a must. Every child has to undergo this operation to get tonsils removed. Every person. And then recently I met with someone. Now the doctors have realized it's an immune organ. You should not remove it unless it is infected. But there they were just proactively removing tonsils.
what is the reason?
Because it can get infected. So that you are removing an immune system organ because it can get infected. It's funny. It's really but now after ages they understand you shouldn't remove it. Okay. Okay. Thymus is the one which helps T cells mature. That's why they get the name T cells. It's right here, thymus. Okay. Bone marrow, we know. Bone marrow is the one which makes all these immune cells. Skin and mucosa, you know how they are protective. So these are some of the cells which are listed here. These are immune cells. Now we divide immune system in two parts. Okay, you know some of the players. So two parts are innate and adaptive. So innate is always ready. The moment infection happens, reaction occurs in that second. Acquired is that they will analyze what kind of infection has occurred. Accordingly, they will make antibodies and then attack. So this takes some time. It takes 2 days, 1 day, 2 days, maybe a little bit more. But this is instantaneous, right? So innate and adaptive. So innate immune system, they don't know what is attacking. They don't have time to analyze it. They just know it's foreign thing. So it's the body's first line of defense, while the adaptive or acquired system takes longer but enables a memory to be created of specific antigens. So suppose we get a vaccine, right? So vaccine, ultimately the antibodies will be made, ultimately, right? And then they will stored in our memory forever. So that is acquired, but the first response is innate to any foreign thing. So innate is present from birth, rapid response, non-specific. It doesn't care what antigen is there, it will just go and try to kill it, okay? And there is no memory involved. This is developed, so here it will break it into pieces, MSC2 will present it, and then they will make antibodies against it. So this is long-term memory here. It's very specific to that antigen. So if we get COVID vaccine, right, our body makes the antibodies and then store it. Okay, so I'm going to show you a couple of these pictures. So this is just showing you, these are the cells which are involved in rapid response, innate, and these are the cells which are responding slow. So B cells, T cells, which make antibodies, this is the slow response. We don't get antibodies instantaneously. It takes time to make it, couple of days to make antibodies there, okay? So this is the same thing again. The reason I included this slide, it shows you the time. So this is instantaneous innate, and this is after 12 hours, and it may extend up to a week. Okay. Now the reason I included this one is that dendritic cells are the guys which is in between, because they are the one presenting it in MSC2, and that's how the antibodies are made. So dendritic cells are the one which are bridging innate to acquired, because they are the presenters. Okay. All right. So they make antibodies. B cells are the ones which make antibodies. And see, helper T cells are informing them here. So B cells have this fail-safe mechanism that they have to double check with helper T cells. And if they say, okay, go ahead, then they will go ahead. Okay? All right. So let's look at innate immunity. So it is Venkatesh, we stop at 10:30. We'll stop at 10:30 wherever we are, and then we'll continue after 15 minutes. Okay? So innate immunity's first line of defense, macrophages, natural killer cells, dendritic cells, because they present antigens to T cells. Okay? They detect abnormal cells and they immediately respond. Now, adaptive immunity, cytotoxic T cells, helper T cells, B cells, right? So they are the one which will make antibodies, and that will have long-term memory. Okay, so cancer cells can evade immunity by reducing antigen presentation. So they are basically messing up the acquired one. They're not even going to present the MSC1 thing there. They are not going to. They just remove the presenter there. They have all the things. They hide it inside their house. Okay, they don't show up. Okay, reduce antigen presentation. On in addition to that, all around it, they will create such an immunosuppressive environment. You can actually connect all of this. You can make an analogy with a human home, and the terror you create outside your home, nobody can come there, right? So, and expressing PDL1. Okay, so they have many ways to evade the immune thing. Now let's, you already know about immune checkpoint, but here is the official thing. Now, right now we were just jumping over immune checkpoints. So why do we even have immune checkpoints? So anytime you get someone really active, there is a chance that the active guy can also damage the self thing. So suppose there is a defect, suppose there is an attack, and B cells are activated and they are making antibodies, and T cells are activated. So once they are too pumped up, when you have too much momentum, right, then you just keep going, you don't know where to stop, and these cells can be very toxic for our own immunity. So that is why the body has natural way of making them quiet. Okay, that is why the immune checkpoints are there. If T cells are active for too long, they need to be quietened, right? They have to become quiescent. So that is why we have these immune checkpoints, to make them quiescent after a while. But tumor cells hijack this machinery, and when they should attack the tumor cells, they make them quiescent. Okay, so those are immune checkpoints. So they occur naturally, but they're misused by those guys. Okay, so the reason why we have immune checkpoints is to prevent autoimmunity, control inflammation, balance immunity. Okay, so act as brake on immune cells to avoid chronic immune activation. So immune checkpoints are mainly proteins expressed on T cells and antigen presenting cells. Okay, so the most important immune cells are T cells and antigen. So suppose I am a T cell, right? And there is an attack and I get hyperactive, okay? But I tell someone that if I'm hyperactive, please stop me, okay? So this person also has a thing as a reminder to tell me, now you have to stop, right? So he's my partner in crime, but he also knows when to stop me. That's what it is. So APC and T cells, they work together, right? But if things go out of control, APC says, enough, you need to stop now. Okay, so these are the, you have already, you know about PD1, PDL1 and CTLA4 very briefly. Today I'm going to tell you in details about CTLA4 because this is used as a therapy for cancer patients. So what happens is that T cells and B cells, they express this protein called PD1. Okay, bear with me. So what are the cells which are talking to each other? APC cells are talking because they're presenting the antigen, and T cells are talking. So T cells, like, as a safeguard, they also have PD1. So if APC cells express PDL1, they lock like this, and they will become quiet. T cells will become quiet. Tumor cells are not supposed to have PDL1, but they will make PDL1 so that every time T cells comes close to them, they will make them quiet. Okay, so they hijack the PDL1. They're not supposed to have PDL1. Okay, CTLA4 is slightly more complicated than that. I will explain you after tea because that will take a couple of minutes to explain. Okay, but let's look at this one here. So this is T cell which naturally has PD1. So this could be APC cell. APC will express PD-L1 and they will become quiet. Tumor cells play the same game. They express PD-L1 and they make it quiet. So how do you get rid of it? You add the antibody for PD-1 or PD-L1. As long as you block one, they cannot lock on, and T cells will remain active. Okay, so that is PD-L1, PD-1 therapy, which is extremely successful in cancer patients. That is immunotherapy. Okay, we stop here because the other one will take a little bit longer, CTL4. So I will stop here for CTL4. Okay, we meet here in 15 minutes. अभी 2 मिनट रुक जाइए. हाँ. So let's start again. So before I show you the role of CTLA4, which is also immune checkpoint, I want to remind you about these T cells here. So remember when I told you about the T cells, there are these CD4 helper T cells, there are CD8 cytotoxic T cells, and then there are memory T cells, but then there are also these tracks. Okay, so now in CTLA4 you will see the role of tracks. Okay, so tracks are naturally present in our body to prevent autoimmunity. Okay, but cancer cells will bring these tracks and basically make the whole tumor environment completely immunosuppressive. So we'll see the role of these tracks, which are regulatory T cells, which are involved in immunosuppression. So now let's see how CTLA4 works. Okay, so so keep in mind, I want to remind you about PD1 and PDL1 again. PD1 is present in T cells, and tumor cells will express PDL1 and directly lock horn with T cells, making them quiescent. But CTLA4 is not like that. Tumor cells indirectly control CTLA4. So what it is? So the way things work is that antigen presenting cells like dendritic cells, so TCR, so they are the one which will present the antigens, right, through the MSC1 or MSC2, right? So they are presenting it, and T cell receptor, TCR is T cell receptor, is recognizing that. That is signal 1. But they don't get activated just by signal 1. They need another signal, and that signal is that CD28, which is expressed in T cell, binds to B7, which is expressed in APC. So suppose you are telling me a very confidential information, I would make sure you are the right person, you are not the wrong person. So you will tell me another code, and then I know, yeah, this is the person I'm supposed to receive the message from. So that is that second signal. This is just to ensure that this two signal occurs, and then the reaction will take place, right? Then the T cell will get activated. However, when the T cells are very, like, all the time activated and they need to become quiet, so they express this CTLA4 receptor. Keep in mind, T cells are expressing this, just like T cell express PD1, they express CTLA4, and now CTLA4 has higher affinity towards B7 than CD28. So if CTLA4 is there, that will bind there, but this is not the signal T cell is looking for. Okay, so when they bind, this is an inhibitory signal. Let me repeat this thing once again, because I want you to understand this thing, and then I will show you how tumor cells are hijacking this thing. So under normal circumstances, when T cells have to get activated, they see the signal from MSC, the peptide signals. Then they also bind B7 on APC via using their CD28. So CD28 and B7 will bind like this. Okay. So when both the signals are there, T cells are going to get activated. After a while, they're very active. They need to become quiescent. So they will express CTLA4. This also binds with B7, but this has a higher affinity towards B7. So if CTLA4 is expressed, instead of CD28 binding this one, CTLA4 will bind there. That has a preference because it is more tight binding. Okay, but this binding is inhibitory, and the T cells, they will become quiet. So now let's say how tumor cells will hijack that. So if tumor cells express CTLA4, they can hijack it, but they don't. They don't. So instead, they recruit TREG cells, the ones which are immunosuppressive cells, T regulatory cells. And TREG cells have high levels of CTLA4. So when they come, they bind with APC, they don't even let the real T cells get to APC. Okay? And basically they don't get the second signal and they are not activated. Is it making sense? So tumor cells are indirectly recruiting TREG cells to inhibit CTLA4 signaling. So there is a difference between PD-1 and PD-L1 and CTLA4. Just to tell you how important it is, there is an oncologist in Delhi, okay? His name is Atul something, okay? So he was saying that in his, he's a specialist in liver cancer. So he was saying that there are two immunotherapies which are commonly given to patients. One is PD-L1, PD-1, along with VEGF inhibitor, right? VEGF means angiogenesis inhibitor. So that is one combination. And second combination, even more aggressive combination, is PD-1, PD-L1, and CTLA4. So he said that that is even more because now you are attacking the immune system using two. So then I asked him, what about VEGF, combining it with VEGF, because you have to also give anti-angiogenesis there, right? So he was saying that that becomes too toxic for the patient, like 3 drugs at once. But he said, in his opinion, combining the two therapies together, PD-1, PD-L1, and CTLA4 is more effective, although it is slightly more toxic to the patient too. Okay, so I'm just trying to tell you how important is this signaling. Okay, so this is what is showing that they recruit TREGs, and TREGs have CTLA4, they bind to APC, and they don't let the activation of T cells occurring there. Okay, so this picture I have included, but this is not a very accurate picture here. So here it seems that it is not showing you that this is actually a TREG which is binding here. Okay, but it is just showing CTLA4 binding here, inhibits the signaling, and if you use the antibody against them, then T cells are active, which makes sense. Because if you put the antibody, then CTLA4 will not get engaged, T cells will get both the signals, and they will become active. So this is a more accurate representation. In conventional T cells, TCR T cell receptor engages with MSC2. See, this is MSC2 of APC. Okay, and CD28 will bind, this is B7 basically. So it will bind here, but if CTLA4 is there, that will take preference if it is there. As long as it is not there, it will bind here, and if it binds here, then T cells will get activated. Okay, but if TREGs are there, they have CTLA4, they will just soup up all this signal and T cells will never get activated. Okay, so now I will briefly tell you about tumor surveillance again, that the immune cells are always surveying all cells. Okay, so this is something that I told you in the very beginning, elimination, equilibrium, escape. So initially they will eliminate anything which is foreign, then slowly some cells will survive, and then the immune system itself will help the tumor to grow because they will quieten the immune system. So now I'm showing you the tumor microenvironment. We have heard of it many times. Now we will actually look at the players of tumor microenvironment. What exactly are they doing? So this is the primary tumor, right? Look at this thing. So this is blood vessels. They need blood vessels, angiogenesis. That is why anti-angiogenic therapy is very effective. Okay, they remodel tissues. Okay, these are remodeled tissue, creates highway for spread. They want space to spread. Okay, so they break up all the net which is there, like really loosen it up so that they can spread. Okay, they get cancer-associated fibroblasts. This is Tregs. These are immunosuppressives. Of course, they will call them. Okay, they make the whole environment really acidic. Okay, so the pH drops all around it. Okay, blood vessels are supplying oxygen to nutrients. Angiogenesis is new blood supply. Now, they are giving, so these are TAMs, tumor-associated macrophages. They are really bad guys. They are also immunosuppressive. I'm going to spend quite a bit of time on TAMs and TRAGs, and also that there is another kind of cells which are recruited. They are called MDSCs. They are also immunosuppressive. We'll spend time on those. So basically, the tumor is surrounded by all tumor suppressive cells. Immune suppressive cells, and that's how the tumors will grow. Okay, so what is the tumor microenvironment is doing? See, look at this thing, MDSCs, these are tumor suppressive cells, immune suppressive cells. TRAGs are the same, TANs are the same. So they recruit all of these cells to suppress the immune system around it, okay? And because of that, the treatment doesn't work either, okay? Plus, it is like you make, around your house, you make nets and nets of defense systems. So such fibrous material protecting the tumor so the drug cannot even come to the tumor, okay? So and obviously they make a lot of blood vessels around it to supply. So tumors are usually hypoxic, low pH, high glutamine consumption. They express PD-L1 to make sure no T cells come to them. Okay, so some of the factors are these: they recruit tumor-associated macrophages. So these immune cells actually help in angiogenesis. Instead of killing the tumor cells, they help in angiogenesis. Okay, cancer-associated fibroblasts, they corrupt healthy cells. They become factories for growth factors for tumor cells to survive. And they secrete all of these factors for their own good. Okay, so very quickly, how tumor cells support how tumor microenvironment supports tumor growth. It supports tumor growth and survival. Let's not go into the details. I have put the details here so that you can always go back and look at it if you want the details, but I'm going to go through the main things only. So the tumor microenvironment is highly conducive to support tumor growth and survival. They secrete all the survival factors to resist cell death and to promote tumor growth. They create an immunosuppressive environment. They recruit immunosuppressive cells. They do two things. They recruit immunosuppressive cells and they secrete factors that the cells which are actually supposed to promote which are supposed to help you, they become immunosuppressive also. It's like you bribe the police to be on your side. Okay, altered antigen presentation. Because this is an inherent thing, they have to present the antigen, they down-regulate their MSC1 and they don't even present, so they become invisible to T cells. They promote angiogenesis, okay. They facilitate invasion. So, these are boundaries there, so tumors cannot, so they break the boundaries. Okay, they change the extracellular matrix, basically break the boundaries so that it can invade. ECM is extracellular matrix. They break the whole thing, secrete factors so that everything breaks apart so that they can spread. Okay, and EMT I told you, right? So when they are brick-like, they cannot move. When they become mesenchymal, they can go through and squeeze through things and they can become very motile, and that's how invasion takes place. Okay, and then they form pre-metastatic niche. So they go and they form new tumors. Okay, they find new places, they settle there, and that leads to metastasis. Okay, so look at this thing. It is invading. Good. Once it goes to the bloodstream, now it can go anywhere in the body, and it can lodge to a new site and then form a tumor there. Okay, and they actually completely change the metabolic programming. So normal cells do not rely much on glycolysis, but cancer cells really rely on glycolysis. It's really upregulated. That's why we do PET scanning, because glucose consumption is very high. Okay, so, and it contributes to drug resistance. Okay, so you guys already know all of these things, so I'm not going to go through this. So basically the tumor, the microenvironment is highly immunosuppressive, completely designed to promote tumor growth. Okay, so this is just showing you all different kinds of things which are going on in the microenvironment. Okay, so one thing I want to highlight once again, That is the antigen presentation. So our body has evolved to always keep an eye on every cell of our body. And like I said, they have to keep presenting the antigens. So whatever is there, you slice them up, you show it. These are the antigens I have. Immune system recognizes as self, let it go. Anything foreign, that cell is killed, right? So normal antigen presentation, so tumor cells will remove the MSC1 completely. If there is no MSC1, there is no presentation. So they don't even show, they become invisible to the immune cells. Okay. So if, hypothetically, if they present it, then T cells will recognize them as foreign and it will kill it. But they downregulate MSC, so they are not affected. Okay. So this is already, you know, I've already discussed this, a tumor microenvironment. And so cancer cells, what will they do? Either they will change the cells to their own good, like they will make them tumor suppressive. So an immune cell comes thinking it will kill it, but they will secrete some factors that they will become pro-tumorigenic and they will support the tumor formation. Okay. So this is what I was telling you, MSC1 and MSC2. All cells have MSC1. So if there is a viral infection and if they present viral infection, T cells will recognize it and kill them. Okay. But APC cells, they are the one with MSC2. So their job is to take the antigen, chew it up and present it. And if it is foreign, obviously, then the T cells will kill them. Okay. So now you know all the mechanisms. So now you can understand what tumor cells are doing. First, they have to make sure that they don't present the antigens because they have unique antigens. So what will they do? Can you tell me what will they do to avoid presenting the antigens? They downregulate MSC, which one? 1. 1 is the one which is present in every cell of our body, including tumor. 2 is present in APC cells only, right? Professional APC cells. Okay, so they downregulate MSC1, can you see there? So that they don't have to present, so they can hide from the T cells. Then they secrete all of these immunosuppressive thing in the microenvironment to make everything favorable to them. Okay, then they do the immune checkpoint thing, CTLA4, PD1, PDL1, you guys know that. Okay, and in addition to that, they secrete all these chemokines to promote angiogenesis, to promote invasion, to break down the extracellular matrix so that they can invade. So now you guys know, right? So they are really making use of the tumor microenvironment there. All right? So initially, when the tumor starts to grow, the tumor antigen are expressed by MSC1 because MSC1 is there and the tumor cells are killed by the immune system. Slowly, they remove MSC1 and they escape from the immune system. So they will first grow the tumor and then they will become even smarter and smarter. And as they completely evade the immune system, the tumor just escapes from the immune system. Okay, so this is the immune cell, this is the tumor cell. Okay. So you don't have to remember anything, but what it is telling you, these are all oncogenes, MYC, RAS, EGFR, PI3K, these are all oncogenes. So how they are using oncogenes? To upregulate PDL1, okay? To upregulate PDL1, immunosuppressive factors in TME, inhibits T cell recruitment. And these are all tumor suppressors, so they delete them. They delete PTEN, 53, RB1, APC, and when they delete it, the outcome is the same. PDL1 is expressed. See, PDL1 is expressed here. BRCA1 is even more higher. So if it is homogeneously high, probably it is not that high, but if some part is really lighting up, then with contrast you can see that it is, very high. Also, when you give radioactive thing, bladder will show up because a lot of radioactivity is leaching from there. Then sometimes kidneys will show up, not because kidney has cancer, but it is trying to get rid of it. So many, bless you, many organs that will show, many organs will show like that. So people who do this kind of reading, they are used to it. So, but you know, the image I showed you was extremely nice. Like, how bad was the metastatic cancer at just one targeted therapy? A person having that kind of metastatic cancer is just really bad to start with, and then one therapy is doing such wonders. So, just VRAP, just one VRAP. Oh, you should use this so that because it. Go ahead. What is the question?
I'm answering to his question. Like I have seen in case of brain tumor cells detecting, they what they do, they give IV which is having some sort of fluorescent compounds, and in case of a tumor cell in brain, that fluorescent compounds just light up there only at the tumor part only,
So if they
and they
told that,
perform the like surgery and all.
yeah. So what happened? No, so for that they have to know exactly what is wrong with that one. So then they can give it. But the good news is that they don't have to do biopsy. They can try different things and they will know what is lighting up. So that is also one way of finding out what is overexpressed, right? And then like folate receptor and things like that, there are some which are generically overexpressed. So whatever is generic overexpressed in brain cells, you can give those also. Give it to, yeah.
Are neoantigens unique to each individual, or there are some neoantigens that are common in every tumor? Like in general, any person have tumor, they will have this kind of neoantigen.
I'm sure there are some common ones and there are some unique ones. So not every tumor will have all unique. There may be hundreds of neoantigens, right? Hundreds of neoantigens. So some will be tumor specific and many of them will be common among tumor cells. Definitely. See, think about it in this way that all the cells in the body has the same DNA, right? Every cell has the same DNA. Even then, all different cells look different, like neurons look different. So a vast majority of the proteins are the same in all cells, right? Because they are the housekeeping thing, they keep the cell alive. And then they are unique proteins which give their unique structures. So in tumors, a lot of mechanisms are the same between different tumors. So those, and which do not exist in normal cells. So those kind of things are going to be shared between different tumors. Or you can say all blood cancers will resemble each other more than solid tumors, right? And then there will be obviously tissue specific antigens. So for CAR T therapy, just one second, the challenge is to find the real right antigen so that it can target there. And the good news is that it's one-time infusion. Once you give the infusion, it stays there for years because they keep on proliferating in the body. But then I did not tell you about T cell exhaustion. So now scientists are working on why T cells are getting tired inside the body and how you can keep them alive, like they don't get tired. That's called T cell exhaustion. Go ahead.
Ma'am, like what mechanism do cancerous cells like use? Like they are like too efficient in terms of mutations. Like they are
In terms of mutations,
yes, like they are keep on mutating. And is this mutations are like randomly, like they keep on mutating, and some mutations are like randomly selected, uh, randomly selected, and these mutations are like the better one survival for them, and they like keep on surviving, or like is it something that they are really too smart so that they can mutate in a way only so that they can restrict the body cells only?
yeah. So the way they, so whenever, so suppose you give Gleevec, I'm giving you a real life example, right? So Gleevec binds in a certain way. So let's say Gleevec has a side chain which binds here, okay, and the residue it is touching is this one. So then what they will do to avoid Gleevec, they will mutate this residue. So they are very smart to know which one to there. So if they add a large hydrophobic amino acid here, Gleevec will no longer bind. Do you see? So there was a threonine there, 315 threonine, which was converted to isoleucine. Isoleucine side chain is bigger than threonine. So cancer cells are not completely randomly doing it. They know that Gleevec is touching here. So if they replace threonine with isoleucine, that will interfere with Gleevec binding and it became resistant. So they are not completely randomly doing. They know exactly which residue to mutate. There may be some permutation where they change threonine to isoleucine, leucine, few others. Those ones survived which didn't bind Gleevec. If they were still binding Gleevec, they will die anyway. So that's how the selection is occurring. But cancer cells are smart that they are not randomly mutating the whole pocket. They are also mutating the whole pocket and elsewise. So some mutations are going on, but they can be extremely precise. So when antibodies are made, right, against any antigen, the way we understand is that trillions of antibody molecules are made, and then the ones which survive are the best ones, right, which are the binders. I don't think tumor cells go to that extent of making those many mutations. They are obviously making many mutations, and it is easy. If you bind Gleevec, you die. We don't need you. The ones which do not bind the drug, they are the ones which survive. So it's automatic selection. So it's midway. They are very smart, but they don't know exactly which mutation to make. So they're making more than one. And only the survivors survive. So these have high, one of the ways they survive is they are undergoing mutation. They are expressing MSC1, then they are not expressing MSC1. They are exposed to something, they do something else. So they are constantly evolving, just like virus. You develop a drug against one virus, the new virus comes in. Cold medicine, every year we get the flu shot, right? Because a new variant comes and the new variant comes and the new variant comes. So cancer cells in that respect are like that. Otherwise, there will be no resistance. Resistance emerging in 3 months tells you that they are mutating fast. Normal cells don't mutate because they are afraid of turning cancerous. So they are happy the way they are. Only they are exposed to extreme conditions like UV radiation or something, then they will mutate. Cancer cells are inherently too plastic, changing all the time. But if you don't apply any pressure, I don't think they will undergo so much mutation. It is under pressure that they mutate. So when you give chemotherapy, they become very resistant. That's when they're mutating to survive. Right? Go ahead.
Ma'am, not every cancerous growth's fate is to metastasize, right? Some can be localized and stay that way for the.
That is true.
So, ma'am, what is there a checkpoint that the cells cross after which they are like they have to do the EMT and have to move away, or is it completely randomized?
No, it is not randomized. So it depends on the kind of cancer also. Some cancers are inherently more aggressive, others are not. Even in the same tissue type, some cancers are aggressive, others are not. So breast cancer is a good example. It has 3 receptors: estrogen receptor, progesterone receptor, and the third one is I think EGFR receptor or something, HER2. So if all these receptors are gone, okay, then the cancer is aggressive from the start. Okay, similarly in pancreatic cancer or hepatocellular cancer, they all have different cancer type. Some are more aggressive than the other. So those cells divide, they are more motile, they are more aggressive, they metastasize. So yeah, it is quality of the cancer, which cells are those and how they are mutating.
So would it be right to say that if you are applying more pressure, there is more odds of metastasis happening, or is it just aggression that is determined by the nature of the cancer that decides what happening?
If you're applying more pressure, the chances are they will die also. But if they are survivors, the survivors will be more resilient, right? But you have to apply a lot of pressure. You have to do everything to kill the cancer cells. But if you're successful in killing it, it is gone. But if you're not successful and they survive, it metastasizes somewhere else. But the thing is this, that we know a lot of people whom we don't know, we just read about them in news. They survive, right? They survive for many years. So we have made a lot of progress.
Ma'am, you told about that a combination therapy like PDL1, PD1, and CTLA4. Like, what exactly is happening in that combination therapy?
So in that combination therapy, so there are two things which are occurring. So cancer cells are upregulating immune checkpoint things to prevent to shut down the immune system. So in some patient, PDL1, PD1 is occurring, and some cases CTLA4. The chances are both are upregulated in patients because the tumor is growing, right? So it is an approved therapy to give PDL1 along with CTLA4. And the oncologist at Max has said that in his experience, when you give PDL1 along with CTLA4, that is better than giving PDL1 alone with VEGF inhibitor. But my thinking is that anti-angiogenesis inhibitor, this is not documented in any paper, any journal. This is his personal experience. But in my opinion, angiogenesis inhibitor is very important. So I would prefer PDL1 angiogenesis inhibitor. I have not seen CTLA4 angiogenesis inhibitor, but that is also possible. But most of the time, if you have to give one immunotherapy, PDL1 is the choice one. So I think that is more commonly changed. But people also, if you check online, if you give PDL1 and CTLA4 together, that's more toxic to the patient also. It has more side effects. So if the cancer is very aggressive, that's when they give that. In my personal opinion, I have seen all the people who are taking immunotherapy, they're getting better. But in literature, it says only 20% people respond. And I don't think so. So it may be in that particular study, 20% people responded. It also depends on the kind of cancer. Immunotherapy is fantastic for blood cancer. And also, there could be other factors also. Person's lifestyle and all those things could contribute to that. There could be other comorbidities there. So every person is different. And I don't think people have done PGX along with immunotherapy. So if they do PGX also, maybe they will find something different from patient to patient to patient.
Okay, ma'am. So my question is coming from APC and T cell interaction, and that too from the second signal, where B7 binds with CD28, and for that inhibition, there's a CTLA4. So what cancer cell do is just the overexpression so that this interaction gets inhibited.
So cancer cells themselves are not expressing it. They are recruiting Treg cells to do that.
Right.
That's the difference, yeah.
Right. So we are using inhibitors of CTLA4.
Yes.
Just in this contrast, if we're using inhibitors, so aren't we blocking CTLA4 will not create T cell hyperactivity?
At that time, you really want to target the tumor cells. So that time hyperactivity is a lesser evil than getting T cells activated, because it is such a immunosuppressive environment that activating the immune system is more important. That's why there is this toxicity issue. Whenever you are giving immunotherapy, there is always a chance of autoimmunity, always a chance of autoimmunity. So this is, doctors tell you in advance, there could be autoimmunity there.
Just to overcome this autoimmunity, do they have different mechanism to counteract this? Because our body is in constant haphazard to manage all the toxicities and all those stuff.
There could be a lot of different mechanisms, right? But quietening the T cell itself is a very important thing. So if you're activating it, I would imagine there is a chance of some autoimmunity. But then also it depends on the dose, right? It also depends on the dose. And then I don't know. So in general, what happens is that T cells, they express CTLA4 only when they themselves think that they need to be quietened. So initially, CTLA4 is not present in T cells. They have CD28 and they have obviously TCR, right? But when they are activated, then that's when CTLA4 is overexpressed to shut down this. So it's like they are themselves putting CTLA4 so that the signal is quietened, right? So I don't know if CTLA4 is mainly present at the tumor site because it is such an immunosuppressive environment. So then it becomes more specific there, right? Because I don't know how many T cells have CTLA4, because once they are activated, that's when they express CTLA4. So if tumor cells are, if T cells are not even activated anywhere in the body, why would they even bind that, right? Because if they were inherently expressing CTLA4, then there will be no T cell activation ever. So only when they are primed already, then they express CTLA4. So now you can imagine that the tumor cells, TREGs, are the ones which are expressing more CTLA4. So the therapy is much better there. And if it has metastasized to different parts of the body, it will be immunosuppressive environment there. So they will work at all those places. I have seen that, tumors which have metastasized in other part of the body, I've seen the tumor disappearing from those places with immunotherapy. Small things disappear. So it is a good one. It's a good one. But looking at CAR-T, it is really good one. CAR-T is really good one.
Ma'am, so as I do daily a question about the oncolytic viruses, my question was that, ma'am, if we are to, what the mechanism of oncolytic viruses is that those will latch on to the tumor cells, insert their DNA, and then ultimately they will proliferate and lyse the tumor cell and expose the antigens to the immune system. So my question is that when the replication process will be happening, the DNA replication of the viral, whatever design genome that we have put in, it will be happening inside the cell, which is an environment with high oxidative stress and all. So won't those replications also be erroneous in some sense? Or would those be conserved?
Viruses, they don't care. Viruses survive because they mutate, right?
Ma'am, but we have put specific things with an intention of their action, right? So what if what we programmed ends up getting modified and the effectiveness of the virus ends up going down?
Worked with virus. So what happens is that when you do virus infection, trillions of molecules come out of the cells. So even if some percentage is mutated, it will not make any difference. Survival of the fittest. The guys which are infecting, they are the ones which are going to survive. The guys which are not able to infect will be gone. So it is going to work for our advantage there. And somebody asked me yesterday about the autoimmune thing. So I checked today. They do not affect normal cells. The autoimmunity is pretty low. Like it's almost negligible there. So we are very good in designing that it only replicates in tumor cells, not in normal cells. So it showed in pictures again and again, normal cells are unharmed. So that seemed like a very cheap option to get rid of tumor cells, right? And you don't even need antigen because you're infecting a virus, and virus itself is clean. Like we do viral infection all the time in our lab. So there are Bosc cells and Phoenix cells which are used to make virus. So when you harvest the virus, the cells actually lies because cells are so sick, right? They are good for nothing after that. They just pop up and release the virus. So the cells are completely gone. So tumor cells are gone. And imagine if virus is infecting. So then the good news is that it cannot even mutate to fix the virus because virus will outbeat them, right? So that seemed like a very good one. So I don't know why it is not used widely. That is something I'm going to check it today, exactly what is going on there. Tomorrow we'll study stem cells. Okay, why we study stem cells is that there are many reasons to study stem cells. With respect to cancer, cancer many times leads to damage to tissue because of radiation, because of surgery. So you can use stem cells to regenerate those things, and that's why stem cells are very important for cancer also. But stem cells are in general very useful. Have you guys studied stem cells?
I've learned about hematopoietic stem cells.
Hematopoietic stem cells, yeah. Have you guys studied iPSC cells? No. Indis pluripotent cells, yeah. Yeah, we'll see all of those tomorrow. All right, guys, next question. Yes, Vanshika.
You told us that when BRCA1 and BRCA2 gets mutated, PARP will perform their function like double-stranded DNA break repair. So, and that is a tough repair compared to single-stranded. So, is it efficient? Like, won't there be any mutations or like mistakes, errors?
There could be some mistakes and errors, but those cells get eliminated, but those which are fixed, they take over. So what happens is that if there is a mistake, those cells will die. But if PARP is able to fix it properly, those cells survive and they take over. So by any chance, if it is incomplete repair, then that cell will die. And the other cells which are fixed properly, they are the one which will take over. So most of the cells will be all right. Yeah. So cells have this bypass mechanism. So, I will tell you something. So suppose you have to, in our lab also we have seen and everyone has seen that if you want to, let me say it this way. Suppose there is a protein A, okay? So you have two ways to find the function. Either you delete that protein A using RNAi, which is a piece of cake, or you add a small molecule to inhibit it, or you add a PROTAC to degrade it, right? So when you do RNAi, so RNAi is relatively new, okay? I mean, it was discovered in late 90s, in 1990s, and then it became a general thing in the lab around 2003 and 4. 5, 6, it became a regular process, right? Before that, it used to take a very long time to get rid of a gene, okay? But even if you use RNAi, it will take 24 to 48 hours to get the protein expression down, right? So 24 to 48 hours is a long time for cells to bypass that signaling. So what if you remove that protein? The cells will use some compensatory pathway to survive. So some cells will die if it is an important protein, obviously, but some cells, however few cells, will find a compensatory pathway and they will survive. So you really don't know the function of your protein because the cell survived, and you think it is not important for survival because you knocked it down and the cells are surviving, right? But if you use a small molecule inhibitor and inhibit it, it is so quick, you add it 5 minutes later, it is gone. And then the cells will die. And then you know, yeah, this protein is really involved because you did not give the cells time to compensate. Like you did not give them 24, 48 hours. When you're using RNAi, they're going slowly down. 100 to 95 to 90 to 85 to 80, slowly, slowly over a period of 24 to 48 hours. So that is plenty of time to for cells to think of an alternate mechanism. Even if they're not cancer cells, they are living cells, right? That's how we live. We are exposed to one adverse condition, we adapt and we adapt and we adapt, right? So, but if you give a small molecule, it is an instantaneous effect. So then you will see the real picture. So we expressed a protein which is supposed to cause oxidative stress. We didn't see oxidative stress because it takes time to express the protein. But when we added the protein directly to get it across the cell, there is a way to do that. Instant oxidative stress, because there they did get chance to, when we expressed it, the antioxidant proteins were upregulated, so we didn't see oxidative stress. But when we added, it was so quick, 5-10 minutes later, oxidative stress increased. So the point I'm trying to make is that cells are pretty resilient. So when you use small molecule versus long process, they will give you oftentimes different results. That is why people use a combination of things, small molecule versus this versus this. Next question, please.
Ma'am, my question was, when BRCA1, BRCA2 are not present in the cell, we give PARP inhibitors to destroy their cell repair mechanism, so they will die eventually. So, ma'am, if the mutation for BRCA1 and BRCA2 is not sporadic but germline, and every cell of the body does not possess PARP, then it would kill the person.
That is a good point. That is a good point. Because every cell will have BRCA1, BRCA2 mutation if it is a germline mutation. And if you give PARP inhibitor, it should affect all the cells. So here is my answer to that. Obviously, people survive. It's a very good drug. So I think normal cells have alternate mechanisms to take over it. Cancer cells are more vulnerable to that. But let me check that. That's a very good point. Actually, when I was teaching you, for a second, this thing came to me. But before I could grab that thing, because I was talking, it slipped off my mind, because I was thinking it is in every cell of the body. But by that time, I was speaking something else, so I forgot that I was thinking. But let me check on that. That's a very good point. That is a very good point. But it is, in general, it is known that normal cells are really efficient in DNA repair because they don't divide as much. So they could have some additional mechanisms there which allow them to survive. Cancer cells are dividing so fast, and that is the backbone of chemotherapy. That because they have defect, even if BRCA1, BRCA2 are there, DNA repair pathway is defective in cancer cells. So they are more prone to damage. They are more prone to damage, and that's why they are more prone to. So, yeah, okay. I will answer your question now. I remember what's going on. So, when you give olaparib or something, you're also introducing DNA damage there by radiation or chemotherapy or something. Otherwise, under normal circumstances, why would they die? So you are introducing DNA damage. So it is often given in combination with some kind of therapy which causes DNA damage.
Told us that in normal scenario too, our DNA is damaged daily in every cell, and it is repaired on daily basis.
That is true. That is true. But there are different kinds of DNA damages, right? So cancer cells are more prone to danger, like double-stranded breaks are very bad. So double-stranded breaks occur when we are exposed to UV radiation, like serious UV radiation or some kind of serious chemical. What happens in daily basis are like deep urination, deep pyrimidation. So it's like, the gums are there. So the gums are phosphodiester linkage. So that is intact, but the tooth is missing. So the base has gone. So they are very easy to fix. Cut it from either side, remove it and add the correct base, seal it. Okay, if there are some other bases, you cut 12, 13 base pair, cut it, fill it with DNA polymerase, ligate with ligase. So these kind of things are piece of cake. When there is double-stranded break, that's where it gets a little bit dangerous. And double-stranded breaks are not happening spontaneously unless you expose yourself to something bad. So if we are sitting in sun for very long, like people do, and sit in beaches, so we may not experience too much damage because our skin is dark, so we have more melanin which protects us. But people who are very white, they lack melanin, and if they sit in the sun, they are more likely to develop skin cancer, right? So these people, so those kind of damages will break both strands of the DNA. So a lot of DNA repair enzymes are mutated in skin cancer, lot of it. So you will be surprised how many cancers are caused by defect in DNA damage, DNA repair. So you can also imagine that it is possible for olaparib, and again I will check it today and then I'll tell you with like really confirm it tomorrow. But it is also, I have seen that they give localized some kind of therapy to introduce DNA damage and then give olaparib, and then those cells will die. Right? Makes sense? Yeah. But that's a very good point. It is in every cell of our body, no doubt about it. But think about chemotherapy, right? Chemotherapy is targeting every cell of our body. We are just hoping cancer cells are more susceptible. So there are bad therapies. I did my part, right? With 4, we'd finished before lunch. Actually, it was easy because you guys had studied a lot of it. Tomorrow it will be all brand new stuff because we have not studied stem cells.
Ma'am, in CAR T therapy, you have discussed about the transvection or the transvectants. What are these exactly, transvectants?
Oh, so what happened? So tell me, has anyone here worked with bacteria and made proteins in bacteria? Nobody. No one.
Cell culture basically, but I haven't
Okay.
been
So
studying
it doesn't matter. It really doesn't matter. You guys look very apologetic and very this thing. Don't worry about it. I don't know the background, so I have to ask to start where I should start, right? So never be apologetic about it, okay? Whatever you are doing is great. It's the greatest what you are doing. So never feel that way, okay? So what happens is that, so let's start with bacteria, right? So do you guys know what a plasmid is? All of you know? Is there anyone who doesn't know plasmid? All of you know, right? You know Evans too, right? Plasmid. Okay, so there are bacterial plasmids and there are eukaryotic plasmids. So what that means is that DNA replicates. So when DNA replicates, DNA polymerase recognizes the origin site, binds there, and then it replicates the DNA and it stops at termination site. So E. coli has different, eukaryotic have different. That's why we have different plasmids. Makes sense? So E. coli is a very good machine to make proteins, human proteins, right? So we take a plasmid and we put human DNA sequence there, right? So, and then we feed it to E. coli. So how do we feed it to E. coli? So there are many ways. You can give them electrical shock. You can buy special cells, E. coli cells. So they are made in such a way that if you get them heat shock, so you put them on ice for 30 minutes, then you take it out and for 45 seconds you put them at 42 degrees or 37 degrees, and then you immediately after 45 seconds you put it on ice for 2 minutes. So they will get heat shock and they will take the DNA inside. So you mix your DNA of interest and you mix it with E. coli cells, not growing cells, these are frozen cells. So you buy these frozen cells or you can make competent in your cells. These are called competent cells. These are nothing but E. coli cells ready to take the DNA inside. So, or you can, it is very easy. They take the DNA inside. Then what you do, E. coli will drop the plasmid. So you put a selectable marker. So those E. coli who have taken will survive in ampicillin. Those who have not taken will not survive in ampicillin. So E. coli is forced to keep the plasmid inside. Now this plasmid encodes your gene of interest, let's say Sarc protein. So this will keep on making Sarc for you and you can purify it from E. coli. So E. coli is a protein production factory for us, for all of us, right? So that process, when E. coli or any prokaryotic cells takes DNA inside, it is called transformation. What is it called? Transformation. Now you can do the same with mammalian cells. You have done cell culture. So mammalian cells will not take DNA if you just add it and you get heat shock. There are reagents you can buy like lipofectamine and there are many of them. So you mix them, put on ice for 30 minutes, then you spread them, then you wait for 1 hour, then you wash and 24 hours later they will express the process protein. So when you do that, that process is called transfection. Okay, so what happens is that the cells will take the DNA, but very few cells will take the DNA. Some cells like to take the DNA inside, so the transfection efficiency can be as high as 90%, but most of the cells will take 10 to 20%. So you will have protein expression of your gene of interest, but not too much. So then also you do selection there. So your plasmid has some selectable marker like puromycin or something. So if you grow the cells in that for a long time, only those cells will survive which have your DNA, right? So you can make a stable cell line. So then there is another way that you take your DNA of interest in a viral vector and then you put it in a virus producing cell line, not your cell line but a virus producing cell line. So because it is in a viral plasmid, it will make viruses and your DNA is inside the virus. Then you take that virus and you add it to your cell. That process is called infection, not transfection. So now 100% cells will take the virus in and you will get huge amount of protein, and your viral vector also has a selectable marker, so you can make stable cell line. So whenever CAR T therapy is happening, they are using viral thing. So in our lab we never do transfection. From the very first day we are doing infection because it is so efficient. You would never do transfection. So that's the difference between transformation. Transformation is in E. coli. Transfection is adding DNA to mammalian cells using some transfection reagent, and then if you make virus of your DNA, encoding your DNA, and then you add it to mammalian cell, that is called infection, not transfection. So that's the difference. So anytime these people are making, they are using viral vectors. So what you do, you take out T cells from the patient. Then for the DNA, you take the DNA for the CAR T cell receptor. And then you package it in a virus, you add it to T cells. Now these are human T cells, right? DNA will never go via transfection. Okay? So you have to use the virus. And then once they become stable there, then you re-inject it to the patient. That's what it is. So I showed you first generation, second generation, third generation. They are just making it more and more and more potent so that there is a more stronger reaction, like weaponizing it even more, right?
Now my question's coming from CAR T cell therapy again. So in a process, we isolate T cells from the patient, we treat them, we either modify them, or in this in between, do we expose them to some sort of antigen during this so that they get modified?
No, 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 no. We already know the antigen before, so we have the DNA which makes the antigen in the same form. So let me tell you something. So what happens is that for nanobodies, for example, right? I will just tell you, even for antibodies. So there is no way that you and I can predict what DNA to make and what, because when antibodies are actually made in human body or in mouse or in rabbit or in camelid, trillions of molecules are made and then nature selects for the best binder, right? So that we cannot predict. Trillions we cannot make, we cannot predict. So what we do is that first we make it in an organism, whether it's a mouse or a rabbit or camelid or something. Then we take that antibody and then we sequence the antibody. So we get the DNA sequence. Now we have the vectors and we have in our labs too. So you can actually produce heavy chain and light chain together, express it in mammalian cells. And once the antibody proteins are made, they will automatically form the proper antibody. Okay. And then you can lyse them up in, there are beads which will bind to heavy and light chains, and you can isolate the antibodies and you can purify them. Okay. So now what you do, that you take a tumor antigen and you raise an antibody in whichever place you want to, like let's say mouse. Okay, you raise it in mouse. Okay, now you know this, then you know the sequence of the antibody. Okay, once you know the sequence of the antibody, then you make it in the lab. So now you know what part will make the heavy chain, what part will make the light chain. So now you keep on mutating it to make it humanized. So you don't change the variable region which binds the antigen, but you change the rest of the thing, right? And then once you do change, that is what humanizing means. You just change it. You are just not touching the variable region. And then that is, you make it in E. coli or some other organism. So this is recombinant DNA. You see, it's not coming out directly from the organism. You're making it in E. coli or some other organism. There are SF9 cells, insect cells, you make these. And once you make them, you purify them, ready to use it. So basically it is coming out of the DNA sequence. Once the nature selects for you, you find out what nature did, and then you make it in the lab. So you guys have heard of AdGene, I'm assuming. AdGene sells a lot of DNA. It is the biggest source of DNA around the world, and it's very cheap. ADDGENE. When I was at ICER Mohali, who is from ICER Mohali? You, right? So I was there in 2016, and I met, there was a lady I met. Of course, I can't remember her name now. I was in touch with her for some time also. So she was getting all the things from AdGene, like, so it is the biggest source of all the DNAs, and it's very cheap. It's not profit kind of thing. So it is like I have given some DNAs to AdGene, so for no cost. So it is like sharing, but they charge you some money because they have to, keep the whole organization going. So it is super cheap. It is just non-profit thing. So the thing is this, that so they sell a lot of antibodies thing. You can make it in the lab. It will assemble it in the mammalian cells or insect cells, and you lyse it up, you can purify it. Yes.
Ma'am, as you said, the neoantigens, there are some neoantigens which are common in all kinds of tumors. So why don't we make a vaccine based on those neoantigens and vaccinate every person. So as soon as a cell mutates towards a tumor, as soon as it grows that antigen, the immune system will
So this
eradicate
is
it.
going to come tomorrow, probably tomorrow, because tomorrow is the second last lecture of the series, right? So there is a topic called immunoprevention which will come tomorrow. So for a lot of things we know already, like HPV virus infects and leads to liver cancer. So you can vaccinate in advance such people so that they are not exposed to liver cancer. So for many viral induced cancer, there are vaccines available. Then they do genetic testing of many people who are more prone to get cancer. So they are also vaccinated against, for which things are known. So it is becoming a common occurrence now that one can get vaccinated for certain kinds of cancer. So our knowledge is not as yet at that we know the new antigen of everything right now. And keep in mind, not every new antigen will really work. You have to find the best guys which are likely to be exposed, right? When the antigen presenting cells are presenting, what they are likely to present. So the way they work is that they take a protein of interest or any protein, they make 8 to 13 mer, right? And then they expose. So not everything will have an immune response. So we have to really figure out what kind of antigen we have to use, which will trigger an immune response. So basically, to make a vaccine, we have to know exactly what we have to use there. So for some, we know what kind of vaccine to make. For others, we don't really know what kind of vaccine to make. Is it making sense? So immunoprevention is a very big thing. So there are vaccines, preventive vaccines. Evans, question.
Okay, my question is about the surface molecules in the cells. I see
Which molecule?
surface
Surface molecules
structures.
in the cell.
Yeah, MHC, the CD, and many
28
others.
and all
Sometimes it
those
is
things.
really confusing to
Yeah, of course.
know
It
which
is
one
confusing
is
to
which.
me
Yeah,
too. So many
yeah,
of them. Yeah.
so many of them. So yeah, so I struggle with that. Knowing CD like MHC one binds with which like MAC8, no, MAC1, then T cell, CD4, yeah, those ones. I struggle with that. Okay, and then my other question is about the viral. This is different from the cancer and inflammation. When they make vaccine, a virus vaccine, when it reaches the clinical levels, how do you test that? Because already you've injected the vaccine to the patient, and you need to see whether it is working in real disease. So for viral infection, how do you test? Because if you inject patients with, say, HPV or any other virus, you risk like infecting the patient with the virus.
Okay, so I will tell you that. So I will know exactly how it is tested also, because I met a person where it was tested. So he was working in his garden, and he was, taking care of the weeds and everything, like, we all have to do ourselves. So he got exposed to some mosquito or some insect, not mosquito, some insect. So he had a very, very strong reaction. It is a very famous thing. I can't remember. It is very common in South Africa also. There is a kind of bee or something, bee sting or something. Like, he had very, very strong reaction. So we went to the hospital, got tested. So they checked for the antibodies towards that. And he had the antibodies towards that because the body's immune system was reacting to that sting. So they knew that. Then they gave him the medicine, and then they said, so he sent me the data to see whether it is working or not. So they said, the report itself said that if you see the antibodies, that doesn't mean it is positive, because once the antibodies are made, they are made. They are not going to subside that soon. So you have to look for any other marker, right? So body is going to make the antibody. So when they give the vaccine, they look for the antibody in the body. Is the vaccine working? Has the antibody been made? Now, to answer your other question, what if the vaccine itself gives the disease, right? Because you're using the virus particle. That is your question, right?
You have vaccinated the patient,
Yeah.
so your intention is to see whether it is preventing patients from being infected by the virus.
Yeah,
Okay.
so you will look for the antibodies against that virus. That is the way to find out. So usually when we get vaccination, nobody comes and tests us that whether we are going to get infected. Nobody tests. We have vaccination, we are supposed to be okay. But if somebody is really following up something, you can look for those antibodies, which is very, very easy to do it. Okay. So what you do is that you take the antigen on a chip or anything like a surface, glass surface, and you take the sera and you add it there. The antibody will bind, of course, if the antigen is there, and then you add a fluorescent molecule which will bind any antibody. So then you wash it away, so only the antibody which is bound to the antigen will remain, everything else will wash off. Then you add the secondary thing with the fluorescent thing, and you will get the signal if the antibody is there. So that is very easy to check that this person has responded. But I, all of us have gotten vaccination. Nobody has followed up on us, right? Whether we 99%, 100% of the time we have antibodies towards that. That is our body's immune response. Now, but I don't know if you asked or someone else asked, what if that the person gets vaccinated? So you don't give the live virus, you give like a tiny bit of piece of a protein kind of thing as an antigen. So there is no chance of, there is no virus there. It is just like a broken nail unique to the virus. So that's not going to cause any damage anyway, but it is going to trigger a strong immune response. So that's the beauty of the body. Just add the tiniest bit in the body. That's how we usually get fever the next day when we get vaccinated and we are sore and things like that. So that means the immune system is working. Yeah.
Does cancer reduces antibodies count also?
Why should it reduce antibodies count?
Because since antibody have potential to kill them.
No, only if the antibodies are going to be produced against the tumor.
But in general, we have antibodies.
But those antibodies have nothing to do with the tumor, right? If we have memory antibodies or anything, they have nothing to do with the tumor.
But we have innate immunity, which consists of IgM, IgA, IgG.
I don't know if the general antibody counts go down. I don't know that. Have you guys heard of the antibodies count going down? I know immune system is suppressed across the board.
Around, but that's the question.
I don't know that. I don't even know that. Yeah, search it.
Serious question.
Yeah. If the antibodies in general go down, I would think that the body is so resilient, the memory antibodies will remain there.
Another question. Right.
By any chance, patients got recovered from the cancer, 100% recovered. Does memory cell hold the memory of, coming back from cancer? Is it?
Unless the antibodies were formed against the antigens. If they were not involved, there is no memory there. But if antibodies were formed, then there will be memory. So if you are killing them by chemotherapy or something, they are not going to form an immune response there. Yeah, so then there will be no memory. Even if you're giving immunotherapy, there is no antibodies involved there. So it's only when B cells find antigens and then they form antibodies and they kill the tumor, then there will be memory there. Otherwise, there will be no memory there. So if you get HPV virus thing, then you are protected from that because you have an immune response. But in general, these therapies, chemotherapy, immunotherapy, they don't involve antibodies. Oncolytic virus, no antibodies. Oncolytic virus can trigger an antibody response and then it will be protected. Then it will be protected. Anytime tumor antigens trigger immune response, keep in mind, maybe, I don't know, it is possible that the tumor microenvironment is so immunosuppressive that oncolytic viruses are killing the tumors, but the immunosuppressive tumor environment is not letting the immune system launch an attack on tumors. So it depends from cancer to cancer to stage. So there are many factors involved. It is like a virus constantly mutating. How is the COVID case right now in India? Is it like once in a while kind of thing? They are not testing.
Gone out of protocol because there was a time where they would recommend a COVID test if you had the symptoms, but I don't think doctors are doing it. I don't think they
Yeah.
have large scale data for the frequencies.
Yeah, it suddenly pops up like in this state the cases are rising, then it shut down.
When people get immunity to that, that's the thing. Because it is not very bad, we gain immunity to that, which is good because self-immunity is good, yeah. But it is constantly evolving, and that's why I am getting alerts, there is a new COVID in rise, get vaccinated. So, I mean, in our area.
And can cancer be used as bioweapon as well?
Say that again?
Can cancer be used as bioweapon as well?
I don't know.
I mean, a nuke would be technically a bioweapon that is inducing cancer. I guess nuke would be a large scale cancer bomb essentially.
But in a micro level.
That would be very insidious if somebody managed to induce cancer with like very small level of impact. That is not external. Oncogenic viruses could be fake.
Oncogenic.
And they could be carrier also, no?
Yeah.
They are carriers if they infect human cells.
They could potentially use
But
as
there are
bioweapon.
oncogenic like HPV is valid.
Oncogenic
but
virus.
the frequency of it causing cancer is pretty low.
I don't know
Because
that.
there's no vaccine against it, and it's pretty curable.
I mean, it's fairly recent the vaccine came out, but
But the thing is, we can create it in a lab. I mean, we can change something in that virus.
you can obviously
it
make the
more,
perfect thing in the lab. Yes, that is true.
more oncogenic and use it as bioweapon.
Very devious.
Discussion going something.
Very devious. Yes.
So, ma'am, what I was going to ask that on the basis of this,
Please, yeah.
on the basis of this neoantigen and antibody interaction, could there be a day when we can use similar tests as we use for viruses, the qRT-PCR kind of test for cancers?
You can even do it today. So, people do biopsy. Okay. So for prostate, they do biopsy. For breast, they do biopsy. I'm sure they do it for liver also. Okay. So biopsy is a must to do. Unless, we feed something like you were saying. Remind me your name again. Kamal. T-A-M? Tamal. Oh, so I never asked your name for sure. Okay. That's very nice name, Tamal. What does it mean?
Actually, I'm from West Bengal, so
Tamal. Is it not Kamal? It is Tamal. Okay. So I am biased, so I heard Kamal. That's what your biasness is, right? Whatever you are used to hearing, you people are biased. People are biased. What do you do, right? I am one of them. So by the time it traveled to my ears, it became Kamal. So Tamal, like you were saying that for brain cancer they give this special thing. So you don't want to go to the brain to do biopsies. It's much better to give a radioactive material. I mean, of course, low dose to scan the brain to see if that is overexpressed. So biopsy is pretty common to find out what exactly is wrong with that cancer. But remind me, what was your question again?
Ma'am, my question was the similar way we were doing for viruses, especially for COVID, we used QRT PCR and we got the positive or negative result in less than 24 hours. So something like that for COVID. So we
No,
could
you can do
act
it.
early.
Yeah, you can do it. So suppose you have the tiniest and tiniest piece of DNA, right, from the biopsy. You obviously have to have something from the tumor cells. For COVID, you can do it from the mouth, but for that you have to go to the tumor cells, right? So if you go to the tumor cell, or BRCA1, you can take a swab and see if BRCA1, BRCA2 is there, right? In like less than, same QRT PCR will tell you whether it is there or not there. If it is a KRAS mutation, you can make the primer towards the mutant site and you will know right away. You can do sequencing. So everything can be done in 24 hours. Every single thing can be done in 24 hours. And you will know exactly what is wrong. And when you say this next generation sequencing, what they do, last time when I came to Gyan course, I actually taught that next generation sequencing also. So you can sequence the whole thing in no time basically without going into details. Okay, so that's what people are doing. If you get a tiny bit of biopsy, you can check the DNA, you can check the RNA, you can check the protein, you can check everything. And it's a very fast turnaround time. So you can do that. The only limitation is that if the tumor is deep inside the body, people are reluctant to do biopsy there. Like I know that person with liver cancer, the doctor would not touch it because they are afraid that they will spread the tumor. So they are waiting for immunotherapy to kick in so that things are under control. And maybe it is possible that hepatocellular carcinoma doesn't have too many targets. So the more generic thing is more common. Like breast cancer, they will immediately go for targeted therapy. If there's some truth is that, and if it is, so not go for chemotherapy. Anyone here? It's a weird thing. You are for. It's a weird thing. You are for since the beginning chemotherapy or since the beginning chemotherapy. He was, but it has to chemotherapy. He was, but it has to the doctor surgically. So the doctor surgically. So.
Ma'am, you tags microbial environment of a tumor is be triggered by the
So not microbial, the stomach.
triggered by the
So not microbial, the stomach, it is too acidic. So make more acidic. It is too acidic. So make more acidic. Will reach the clinical.
reaches the blood-brain stomach reaches the blood-brain stomachs which are releasing the acids
When vary the compound.
form blood is
When vary the compound.
getting pure
So it did go to the brain unless.
getting pure
So it did go to the brain unless. CL will still reach. CL will reach the animals and animals with visible tumors. So the clinical trial in that. And animals with visible tumors. So the clinical trial in that.
What did you mean?
That.
What did you mean affected? I think the stomach's yeah.
It is not there. It is in the west stomach which has it, but so
Be affected. I think the stomach's yeah.
it is not there. It is in the west stomach which has it, but so blood is not that. I have not seen kind of thing. The that even if you forget that thing, if you have the background, you can always pick up any 10 steps of glycolysis and 8 steps. You can very easily connect to anything, and you will at least know what question to ask there, right? Easily, right? If you give them something, they will make a connection out of it. That's the best way to put it. You put 3 things there, and 3 things do not exist in it for the same question. Yeah, it is very
Slow down our learning process also.
yeah. People, like students, are the first one to know all the beautiful things. So he did not come because he was an international student, and don't think I will come back again here. So let me stay here. So he didn't like corrected it, right? And I thought it was a wonderful GPT wrote for him. But he was, so I said, it was always hard for me to make, so right, I have to work hard to come up with 100 questions for you in a second. Yeah, it is very useful because it can give you answers. If you know enough, then you can see, right? Oncolytic virus, are they checked?
Do the antibody count go down?
So what is the situation? One of the big thing, that's why there is so much money eating preservatives and tin food. This is Alzheimer's disease because people have much longer life, because these people live longer. Even so, you hope that the oxidative stress is the root cause for that thing, that it prevents a lot of diseases. That's very good. All of us need that. Your rest of the day goes so well. I like that you are using so many colors. It is reminding me. That is a very good thing. Yeah.
Ma'am, I had asked you the question about tumor cells. I think I might have found the answer. I just want to confirm with you.
Yeah, absolutely.
So could it be possible that the ROS damage just does not get it to in time? So that
Learn. Okay, I don't think 202 on something, right? So viruses are rapidly dividing, and it is there for like years and years. I don't think oxidative stress affects these guys that much. Same thing with E. coli. If you pour it on us, right? They are lot less Alzheimer's disease here. And it is partly, oftentimes we make fresh food, right? We don't use preservatives. So all of these things add up. Heard of it? You have not heard of it, right? So see, every second person you hear with cancer. And air is so clean. Air is so clean, no pollution, no infection. You have annual checkup, prevalent. Part of it, people are stressed all the time. You have everything in life and you still get stressed. And talk off the record. Thank you. Weaponizing things. I was saying, don't say that. As a student,